Good morning.
Good morning, ma'am. Good afternoon, ma'am.
Sorry, it's already afternoon. Good afternoon. Okay, it's 27. Okay, so let's start. Only 28. What happened? Only 28 of you are present today. Is it because I informed you late for the class? Today's class is important, okay? I wanted that everyone attended this class, but it's less. But anyway, let me start my class. So we were discussing, in the previous class, we were discussing about the secretions from the stomach, right? So we discussed, is my slide visible? Okay. I cannot do slide presentation. Okay, so we discussed about the secretion from the parietal cell. The secretion from the parietal cell is the, what was the secretion from the parietal cell? What are the secretions from the parietal cell? Anything else? From the parietal cell, in addition to ACL, there is secretion of a factor known as intrinsic factor. Okay, you remember this, okay? The types of the glands and the glands present in the different positions, different portions of the stomach, and the specific gland, name of the gland and their secretions. Okay, in addition to ACL, the parietal cells secrete its intrinsic factor also. Okay, so any factor, anything which is going to stimulate the secretion of parietal cell, okay, they are going to stimulate the secretion of ACL as well as intrinsic factor. You remember this point. Okay, so we have discussed about the secretion of parietal cell, the parietal cell secrete, and we have discussed how the ACL is secreted by the parietal cells. Okay, and next point, we are going to discuss about the secretion of the chip cell. Chip cells, okay, the chip cells, particularly in the fundus of the stomach, secretes a proteolytic enzyme. Here, I'm telling a word proteolytic. What do you understand by the word proteolytic?
Breakdown of proteins, ma'am. Breakdown of proteins.
Break down the proteins, means these enzymes will break down the proteins. So they are called proteolytic enzymes. Okay, one proteolytic enzyme is secreted by chip cell in its precursor form. precursor means before it becomes an active enzyme, okay, it is secreted in its precursor form, that is pepsinogen, okay, into the lumen of the gastric gland. So now do you understand the meaning of the lumen of the gastric gland? Imagine that this is one gland, okay, this is one cell in one gland. The gland is one whole gland like this, going inside of the mucosa of the stomach. Okay, the gland is going to the inside of the mucosa of the stomach, and the gland is having different cells. Here we are talking about the chip cells, okay, the peptic chip cells. So these cells, they will be secreting their secretion, a proteolytic enzyme, to the lumen. Okay, lumen means here. Okay, say like if we enlarge, so this is, you just imagine that this is one gland and this will be secreting its secretion into the lumen. Okay, so the chip cells in the fundus of the stomach secretes a proteolytic enzyme in its precursor form, that is called pepsinogen, and this secretion comes into the lumen of the gastric gland. The enzyme is secreted in inactive form. Why? Because if it is an active enzyme, right after secretion, it will act onto the surrounding cells. Got the point? After the secretion comes to here, and if it is active enzyme, it may act onto the protein layer of the cells, because cells, all the cells are consisting of the protein, so they may attack onto the protein walls because they are proteolytic enzymes. So to prevent that, this enzyme is secreted in the inactive form. Okay, so this is secreted in the inactive form to avoid digestion of the chip cell, which might occur if it was in an active state within the cell. Okay, so it may attack itself. So to prevent that, they are secreted in the inactive form. Pepsinogen is then cleaved to active enzyme pepsin. That means this pepsinogen will be bigger in size, and it will be from this pepsinogen, some portion will be cleaved, means it will be cut. Okay, it will be cut, it will be clipped, that portion will become the active enzyme, and that enzyme is pepsin. And this activation will happen in the presence of HCl. Okay, or maybe, or by the presence of the already formed pepsin plus hydrochloric acid. Okay, the presence of the pepsin, already converted pepsin, having some amount mixed with HCl, they will convert the pepsinogen into pepsin, and now the enzyme will become pepsin. So if we talk about the size, pepsin is smaller than the pepsinogen. Okay. Chip cells of neonates of cut chewing animals also produce a proteolytic enzyme known as renin. Okay, remember this the spelling R-E-N-N-I-N. This enzyme is different from that enzyme R-E-N-I-N. R-E-N-I-N, renin, comes from the juxtaglomerular cells of the kidney. But here it is R-E-N-N-I-N. This renin comes from the chip cells of neonates of cud chewing animals. Cud chewing animals means particularly the ruminants. Okay, so this renin, it helps in coagulating the milk in the stomach of the neonates of cud chewing animals. Because you know, if the milk is not coagulated, it will easily pass on to the successive portions in the digestive tract. That means instead of remaining for longer period in the stomach, it will be easily passed on to intestine, that from intestine to the different portions of the intestine. Okay, so to prevent that, the milk which is liquid, it needs to be coagulated, means semi-solid form, okay, inside the stomach, and that is done by the enzyme called renin. Renin is secreted by the chip cells of the neonates of cud chewing animals. Okay, so renin, another name of renin is chymosin. Okay, the chymosin is another name of renin. So renin curdles or coagulates the milk in the stomach for digestion, and if it is not coagulated, the uncoagulated milk will rapidly pass the stomach. Okay, so there will be no action of pepsin onto the onto the protein. So that protein will not be digested. Okay, instead it will go directly to the stomach. So in the stomach, the action of the pepsin will not be there. There will, in the intestine, there will be no action of the pepsin. There will be no action of the HCl. Okay, so in that case, the digestion will not be proper. So to prevent that, in case of the younger neonates of the cud chewing animals, there is secretion of enzyme renin by the chip cells. Okay, so here also, what are the secretions of the chip cells? Chip cells secretes pepsinogen in the form of chip cells secrete pepsin in the form of pepsinogen, and which is activated in the presence of ICL. ICL will be coming from the parietal cells, and also the presence of some amount of already activated pepsin mixed with ICL will activate more and more of the pepsinogen and convert to pepsin. So now this pepsin will be ready to act onto the proteins present in the diet inside the stomach. Okay, so we have discussed about the secretions of this. So next, another point is, who are the factors who will regulate, who are the factors who will regulate the secretion of these cells. Okay, first thing we'll discuss about the secretion of parietal cell. Who will stimulate the secretion of the parietal cells? So there are mainly three factors. The first one is histamine. Okay, histamine. Do you remember what is histamine?
Anticoagulant.
No, it is not anticoagulant. Anticoagulant is heparin.
Histamine is vasodilator.
It is a vasodilator. Yes, it is an amine. Okay, and this is secreted. Okay, so this histamine is secreted by certain cells, okay, in the body. So whenever there is more amount of secretion of histamine, particularly histamine secretion increases during allergy. Okay, so but there are again two types of histamine receptors. Okay, so which acts during allergy and which acts during this secretion of this ICL. So that is little deeper, so I will not go into that part. So this Okay, I was telling about histamine. So histamine, whenever there is more secretion of histamine in the body, okay, so the histamine from the enteroendocrine cells in the base of the glands, okay, so whenever there is more secretion of this histamine, the histamine will act onto their receptors. Okay, fine. So histamine, the gastric glands are also having histamine secretion. It is not only the other tissues, right? You remember the other cells in the gastric glands. So histamine is also one secretion, right? So one secretion coming from the enteroendocrine cells in the base of the gastric glands. So that histamine will bind to their receptors. Okay, so the histamine producing enteroendocrine cells will produce histamine, and these histamine will bind to their receptors. So I was telling about the two types of the receptors are there. One is histamine H1 histamine receptor, and another is H2 histamine receptor. Okay, the one which is associated with allergy is H1. Okay, the one which is associated with ACL secretion is H2 receptor. Okay, so this histamine will bind to the H2 histamine receptor. Okay, so during this binding, okay, so because of this binding, this will activate, okay, this will activate the, what will happen? This will activate more and more of the pumping of the potassium inside and more pumping of chlorine outside. Okay, so this part we have discussed earlier. So due to this, apart from this, okay, apart from this binding, what will happen? The histamine producing enteroendocrine cells, they have receptors for another hormone, gastrin. Gastrin comes from where? We have discussed this. From where we get the gastrin?
Peptic cells.
From?
Stomach, peptic cells.
From the enteroendocrine cells, okay, in the pyloric region, okay. Gastrin from the enteroendocrine cells in the pyloric region, okay. So that gastrin secretion will increase when there is more distension of the pyloric region. That means when food is accumulated in the stomach and when it is about to pass the stomach and go to the intestine. That means the food is passing through the pyloric region. So when the food is present in the pyloric region and when the pyloric region of the stomach is distended, due to the distended portion of the pyloric region, there will be secretion of gastrin. Okay, so this gastrin will be available now to bind to their receptors present on the histamine producing enteroendocrine cells. Okay, that means when more of the distension is there, there will be more gastrin production and there will be more binding to the gastrin receptors present on histamine producing enteroendocrine cells. Okay, that this in turn will increase secretion of histamine. Okay, not only this, the gastrin, okay, gastrin which is produced from the enteroendocrine cells due to distension of the pyloric region will also find their receptors present on the parietal cell. So this is showing the parietal cell. Okay, so their receptors are present on the parietal cells, and due to this also there will be more pumping of potassium in and more pumping of chloride ion outside. Water is already present, and due to that there will be presence of the hydrogen ion also. So with that there will be more and more production of HCl by the parietal cell. Okay. In addition to this, the parasympathetic stimulation, okay, the stimulation of the parasympathetic branch of vagus nerve. Okay, so vagus nerve is showing here, is shown here in the form of this green line. Okay, so a branch of the vagus nerve terminal terminating onto the parietal cells. They will be producing acetylcholine. Okay, the parasympathetic part. Okay, so the post-ganglionic nerve terminal of the parasympathetic nervous system will secrete acetylcholine. You remember? Do you remember? Yeah, you remember that acetylcholine is the neurotransmitter secreted by the postsynaptic or post-ganglionic nerve terminal of the parasympathetic nervous system. So the acetylcholine will find their receptors present in the parietal cells, and they are called the muscarinic receptors. Okay, so due to these three actions, there will be more production of HCl by the parietal cells. Okay, so apart from that, when there is stimulation of the vagus nerve and that also the parasympathetic branch, okay, there will be secretion of acetylcholine, and which is also going to stimulate the gastrin production by the pyloric region. Okay, so a branch of the vagus nerve is also reaching to the pyloric region of the stomach, and that will also lead to the secretion of more of gastrin. Okay, so whenever there is parasympathetic nervous system stimulation, there will be more HCl production. Whenever HCl production through acetylcholine release, through gastrin release, and gastrin acting onto the parietal cell for HCl secretion, gastrin acting onto the histamine producing enteroendocrine cells for producing more histamine. More histamine production means more of the HCl production. Okay, so there are mainly three factors, and they are correlated to one another. Okay, so I just read all these lines. If anything was missed while explaining, you can cop up with that. Okay, so first one, the factor stimulating secretion of parietal cell is the release of histamine from enteroendocrine cells in base of the gastric glands. Okay, one. Another is gastrin from the enteroendocrine cells in the pyloric region due to distension of the pyloric region, or due to rise in HCl, rise in the pH in the pyloric stomach. Okay, so whenever there is rise in the pH, that means acidity is low. So when there is less amount of acid in the pyloric stomach, then also is going to stimulate the gastrin production. Okay, so one is histamine, then another.
Excuse me, ma'am.
Yes.
Ma'am, someone is trying to join, ma'am.
Done? Is it done?
Yes, ma'am, done. Thank you.
Okay. Okay. So the second one is gastrin, and another is, the third one is stimulation of parasympathetic branch of vagus nerve, releasing acetylcholine. And in addition to stimulating the parietal cell, the vagus can also stimulate gastrin secretion. So that what you can think of is, when more and more of the distension of pyloric region in the stomach is there, that means when more of the food is present, there will be more stimulation for gastrin production. Okay, so gastrin production is going to stimulate histamine production. Gastrin production is going to stimulate ICL production. When there is more of the histamine, it is going to stimulate ICL production. Okay, and gastrin production will increase as and when there is distension of the pyloric region, or there is fall, when there is rise in the pH, that means fall in the acidity. Okay, so all these factors are responsible for stimulating the secretion of parietal cells. I think you will be able to explain it. This is important. Okay, you remember this. This is an important topic. How the ICL secretion, you can also, how the ICL secretion is stimulated. You can also correlate to your way, how you feel after eating large amount of food. Sometimes we have acidity and all. Why? You know, because there is more amount of the distension, there is more secretion of ICL, more secretion of gastrin. That leads to more, because more is the food, more will be the ICL secretion, more will be the pepsin secretion. That is the need of that body. Okay. Then another point is, who will inhibit? Who are the inhibitors for gastric secretion? Who will be the inhibitors for gastric secretion? So one very important point is, when the acidity of the gastric juices increases with pH below 3. That means when the pH is already low. Got the point? What I said just now, gastrin secretion increases when the pH rises. Okay, so when the pH is already low, the gastrin mechanism for, yes, is it done? Let them try again. I didn't get any response. Who was trying? Is it done now? Two have come. Okay. Okay. So we were discussing who will be the inhibitors for gastric secretion. Who will inhibit it? That means, you know, like, it is not that when it is once stimulated, it will keep on secreting and secreting ICL. Someone has to regulate it. Otherwise, there will be higher and higher acidity, isn't it? So this mechanism needs to be regulated. So who are they? Who will regulate the secretion of ACL, when the acidity of gastric juices increases with the pH below 3, the gastrin mechanism for stimulating gastric secretion becomes blocked. Okay? Is it okay? Okay. Do you agree here? Do you agree?
Yes,
Yes,
ma'am.
ma'am.
Yes. Say, I told you that whenever there is distension or whenever there is rise in the pH, there is more secretion of ACL. But when the pH is low and when the pH is already very low, when the environment is already acidic, then gastrin secretion will be inhibited. So the gastrin stimulating ACL secretion is not required at that moment. So at that time, the gastrin mechanism for stimulating gastric secretion becomes blocked. So this effect results from two factors. One is greatly enhanced acidity depresses or blocks the secretion of gastrin itself by Z cells, or another is the acid seems to cause an inhibitory nervous reflex that inhibits gastric secretion. Okay, so there is also some likely chance that there is reflex inhibition, nervous reflex inhibition of ACL due to low pH. Okay, so one factor here. Another second factor is cholecystokinin and several other hormones produced in the duodenum have a minor role in inhibiting acid secretion. Also means to some extent cholecystokinin, okay, this will be the cholecystokinin will respond in cholecystokinin secretion will increase in the presence of proteins or fats. Okay, as in when the proteins and the protein materials or the fat materials are present in the duodenum. Okay, this is going to stimulate the secretion of cholecystokinin. Where from that protein or the fats will come? The protein and the fat which is present in the partially digested food which has just reached from the stomach to the intestine, to the duodenum. Okay, so here when there is presence of some amount of food, okay, there which is having protein materials, which is having fat materials in the duodenum, it will lead to cholecystokinin secretion. Okay, so now this cholecystokinin secretion and to some extent some other hormones which are produced in the duodenum, they also seem to have some amount of inhibitory action on acid secretion. Okay, another hormone, secretin. Secretin is a hormone synthesized in the duodenum in response to low pH. This hormone does not inhibit parietal cell acid production. Okay, this hormone is having no effect, but their secretion, the secretion of secretin occurs in response to low pH, but this secretion is not having any role. Okay, so who are the inhibitory factors? One is the lowering of the pH itself that may happen through gastrin mechanism or through nervous mechanism, through the nerve reflex, and one is the cholecystokinin. Okay, and other some hormones which are produced in the duodenum. Okay, is it okay? So what?
Yes, ma'am.
Yes. Hello. Anything? It's okay. Okay. So we have discussed about the secretion of the chip cell, who stimulates the secretions of the parietal cell, that is acetic ICL secretion, and who inhibits the secretion of the ICL. Okay. So the secretion of ACL is assumed, okay, particularly it is assumed in case of the human beings, but it can be applied in the other animals also, okay. So this gastric secretion can occur in 3 phases. You can remember like this, that can occur in the 3 phases. One is the cephalic phase. The cephalic phase is when the animal sees of the presence of food or thinks about the food, or the smell is reaching to the animal. Okay, so that by that time also acid secretion starts. So that phase is considered as a cephalic phase. Okay, and another phase is the gastric phase. Gastric phase is when food is present in the stomach. Okay, another is the intestinal phase. The intestinal phase means when the food is present in the intestine, when it is present in the duodenum, and which stimulates the secretion of cholecystokinin, the secretion of secretin, and many other hormones. Okay, so that are also going to lead to the secretion of the ICL. Okay, so if somebody asks that, you can say that it happens in 3 phases. One is cephalic phase, gastric phase, and the intestinal phase. And who are the stimulators? You can explain with the points which I have given. You better note down. These are not in these few points. You better note down. These are not in there in your notes. You can note down this. Do you want to note down these 3 slides today?
Yes, ma'am.
You note down these three few points.
Yes, ma'am.
Slides. Okay. Or you can also make a small sketch. Just put the name of the hormone here. No need to make very good sketch and all. Just put histamine over here and receptor, histamine receptor here, gastrin over here. Just write gastrin here. Then gastrin receptor acting onto the gastrin. Then you just write acetylcholine acting onto their receptor, like that. Okay, and you make this as a like these substances circulating in the blood. Okay, so you can start from here, from the first slide. So you can note down this.
Yes, ma'am.
Is it done?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Copy this.
Yes, ma'am.
Okay, ma'am.
Is it done?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Next. Next, this one. थोड़ी देर में आना क्लास से. Is it done?
Yes, ma'am.
Okay, and then the phases for secretion.
Yes, ma'am.
Phases, you write like this, okay? And on this heading, you make it different phases of gastric secretion. You can make 3 phases. One is cephalic phase, gastric phase, and intestinal phase, like that, the 3 phases. It is there already in your notes. So this diagram is just for the explanation. So you just note down that there are 3 different phases of gastric secretion. That is cephalic phase, gastric phase, and intestinal phase. Okay. Okay, that's all for today. You read it, okay? You read it. You can refer any book, or you can refer to, I have referred to the Duke's Physiology of Domestic Animals, the soft copy I gave you. Okay, I have referred to that. You can refer to that book or any other book. There is no problem, but you have to read it. This is a very important topic. Okay, how they are regulated. Okay, if it is done, then you can leave. Okay.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Welcome.
Thank you, ma'am.
Thank you, ma'am.